Right. 1.2 Structure and function of proteins. Now, let's start with learning intention. Protein synthesis involves transcription of gene into messenger RNA and translation of mRNA into an amino acid sequence, also known as polypeptide at the ribosomes. Transcription and translation. So, what we are learning in this unit is the central dogma of cell biology. The word dogma means principle, and the central dogma we'll be learning here is basically two steps. First, transcription, and after transcription is translation. Transcription is the process of turning DNA strand into mRNA strand, and that happens in the nucleus. And translation is when you take that mRNA strand and you translate into protein. This happens in the cytoplasm at the ribosome because we already know that ribosome makes protein. In this unit, we're going to discover more deeply how ribosome does it. Okay, so without further ado, let's get to understand two terms, coding strand and template strand. First, we need to know that DNA has direction. We already know that DNA is a double helical structure. That means it has two strands, and one of those strands runs one direction, say up. If that's the case, then the other strand will be running the other direction of the first strand. So there's a word to describe this relationship. It's called anti-parallel. This means that both strands are parallel, but they're just running in opposite directions. So in a DNA strand, there's always going to be a coding strand, and the other is always the template strand. The coding strand contains the desired sequence for the making of protein. So the coding strand is really the one that you want for the synthesis of mRNA. Or simply put, you can imagine it as containing the right information, whereas the template strand doesn't contain the desired information for protein synthesis. And we'll get into why one is wanted and the other is not wanted. Okay, so you have the coding strand, that's the one you want, and you have the template strand. For example, up here, right? We have the non-coding strand on top, and you have the coding strand. An mRNA is made when spare nucleotides attach to the non-coding strand because due to contemporary base pairing rules, we know that anything that pairs up with these bases is always the opposite strand. So when uh, one, an mRNA wants to contain the information from the coding strand, it attaches to not the coding strand, but the template strand, because template strand contains the other side of the coding strand. So as a result, the mRNA made from bonding to the template strand will actually be information from the coding strand. That's why it's called the template strand, because mRNA uses it as a template. Next, we're going to talk about translation. Translation is the translation of mRNA to protein. mRNA is a single strand of RNA, and it is responsible for protein synthesis. So, tRNA carries amino acids to ribosomes. Every 3 nucleotides in mRNA code for 1 amino acid. Amino acids are joined to form long protein chains. And here we need to be able to draw a process of translation. Okay. Now imagine that we start with the DNA, and the DNA triplets in this strand is TAC, AAA, GCG. Now, what an mRNA will produce from this coding strand is AUG, CGC, because an mRNA would have uracil instead of thymine. Now, a tRNA is right here. tRNA matches up with the codons, and they are called, the tRNA are anticodons. And at the end of each tRNA carries amino acids. So you will literally have 3 nucleotides pairing up with 3 one. So you have 3 nucleotides in one codon pairing up with 3 nucleotides in one anticodon. And the information from the mRNA would code for the sequences of amino acids. And the sequences of these amino acids matters to the property, the shape of the protein produced. So, because these two then bond together, we know that tRNA must be the opposite side of mRNA. Therefore, this tRNA anticodon will be UAC-AAA-GCG. Now, a tricky thing that a lot of people confuse with is the last step, often the protein amino acids. So there are about 20 different protein amino acids that are named. And you will see basically a list of these AUCG letters and their corresponding protein amino acids. But it's important to notice that these protein amino acids, these names are based off not the characters from the anticodon, but from the characters of mRNA codon. So that's kind of weird, right? Because following this diagram, you would naturally think we get these letters from the anticodon. But no, these letters are based off AUG. So if you see AUG, you would know that its corresponding amino acid is MET. But if you see EAC, if you think that MET is the amino acid for anticodon, UAC, that would be an incorrect answer. So in summary, transcription happens in nucleus. It is DNA to mRNA. And translation happens in cytoplasm in the ribosome. And it's the translation of mRNA to protein. DNA is double-stranded, has bases adenine, thymine, cytosine, and guanine. They are long strands, and they have one less sugar than RNA, and that's why they're called deoxyribose nucleic acid. mRNA are single-stranded, has bases adenine, uracil, cytosine, and guanine. They are shorter segments, and they have that oxygen that DNA doesn't have. But it's more complicated than this. There are some more extra steps in it. Now we need to learn about exons and introns. Exons are segments of a gene that code for synthesis of a protein, and introns are segments of a gene that do not code for synthesis of a protein. Yeah, I misspelled some letter here. And there is pre-mRNA, which contains both introns and extrons, and there is mature mRNA, which will be translated. Okay. Now that's everything from the part A. Because I didn't include many diagrams in my PowerPoint presentation, some parts might be very unclear, such as exons and introns, but I try to explain this as clear as possible. So we code for mRNA, right, from the template strand of DNA strand. that we had earlier. A mRNA strand will literally pair up with the bases in the template strand. So if you have A, T, C, G in the template strand, your mRNA bases would be a U, T with A, C with G, and G with C. So it works something like that. So you have a short segment of mRNA, and this mRNA, despite it's short enough, it still contains exons and introns. Intron is spelled some letters here. And you then have what you have now is pre-mRNA, but that's not the final mRNA that codes for your protein, that codes for the sequences of your amino acids. That same segment of mRNA is cut short, so the introns are cut out, which left the mRNA becoming a mature mRNA, and this will be translated into proteins. Very intention of part B. 1.2.4 The folding of a polypeptide to form a protein with unique three-dimensional shape is determined by its sequence of amino acids. Now, this is something we have mentioned earlier, why DNA is basically responsible for the, uh, the synthesis of protein. So if you mess up your DNA, your mRNA will be messed up, and as a result, your protein, the amino acid sequence, will mess up. And because protein is a big part for functioning of our body, if you mess up the protein, you're basically screwed. And that's why DNA is important. You don't want to mess it up. You don't want to go to Chernobyl and mess up your DNA because that's how you die. Next, we get to know the four structures of protein. There are four structures, and they are in order primary, secondary, tertiary, and quaternary. They're kind of weird, but I'll explain them one by one. Primary coding structure is the sequence of the chain of amino acids. So amino acids are just made from three codons or three anticodons. And there are 20 of different types of amino acids. And these 20 amino acids put into different sequences will give you different proteins with different structures and different functions. And if you zoom out a bit from the primary protein structure, you'll get to secondary protein structure. Now, they come in two types. There is pleated sheet and alpha helix. Pleated sheet is literally a sheet shape of protein, of many of these. And alpha helix is just one helix structure of many of the amino acid chains. And they form because when the sequence of amino acids are linked by hydrogen bonds. And when you zoom out a little more, you'll get to tertiary protein structure. This occurs when certain attractions are present between alpha helix and pleated sheets. So these are a mixture of these two. It gets even complicated here. But this from now on doesn't really look like its structure, doesn't really look like it has a specific design to it. It just looks like a messed up wool, if you know what I'm saying, like your earphone that is constantly messed up when you place it in your pocket. Or tangled up, doesn't have a shape, just messed up, right? But actually, it's very condensed in a very complex way to make it store a lot of information, to make it do what it does. And when you zoom up a little more, you get to the quaternary protein structure, which is a protein consisting of more than one amino acid chain. And this is pretty simple. This is basically if you take two of the tertiary protein structure and put them together, basically one them up together, and you get a quaternary protein structure. Next, we go zooming to on each one. Primary structure. So the little round balls you see on the chain is amino acids, and this is the amino acid chain. Read the primary protein structure is the chain of amino acids that makes up the proteins. The key is the sequence. So the sequence that these balls are placed in matter to the function of the protein that is produced. Pretty amazing, right? You can just place this in a specific order, and the protein produced will actually form different structure because of the way that the bonds work. And then the secondary structure. Remember, a secondary structure has two types, pleated sheet and the alpha helix. This is a bit complicated. This is showing all the the real chemistry going on behind it, but I don't think we need to know this for biology. Next, tertiary structure. Now, this is getting to what I was saying earlier, why the bonds matter. Why do proteins have different shapes? Well, because the hydrogen bond that's pulling them together can work differently. And when they work differently, it gives them different shape. If they bond at a specific place and not others, it could give this protein specific function or specific shapes. But it's really complicated, and we don't really get into this in year 11. And you don't really need to know any of the labeled words on this diagram. And finally is the quaternary structure. Now we get into something interesting, ricin. If you've seen Breaking Bad, you will be more than familiar than to what ricin is. Ricin is what Walter White used to poison many people. It's really poisonous, and it basically messes up your protein. I think doesn't say here, but ricin basically mess up your protein synthesis, right? So we already know that your body depends heavily on the proteins you produce. Protein plays a big part in your body. If your protein gets messed up, you are gonna die, simply put. Now the quaternary structure is basically two or more of tertiary structure winded together. The key is the specific 3D shape of the protein is its specificity. If you change the specific 3D shape of the protein, you are likely to stop it from functioning. Or in terminology, in biology words, if you change the specificity of the protein, you will probably stop it from functioning. And finally, we get to the learning intentions 1.2.5. Proteins are essential to cell structure and functioning. Again, this is the real reason why proteins are important. They are essential to cell structure and cell functioning. Examples of proteins with specific 3D shapes include enzymes, some hormones, receptor proteins, and antibodies. And that is the last slide in this PowerPoint presentation. Well, thank you. That was 1.2. Thank you for studying the unit with me. I will also work to add more diagrams like this into my presentation since now it's really vague and heavily based on words. So if you're a first-time learner, this may require a bit of imagination, but I'll work to add more images in the presentation, make it clearer for you guys. Well, thank you and have a good day. Enjoy biology always. Peace.